Well, if you thought you had fun with cellular respiration the first time around, wait till you see this video. This one will go ahead and give you a lot more detail concerning the process of cellular respiration. Now, if you want extra credit and you're a bio student, have at it. If you're an honor student or an AP student, then yeah, you should probably definitely watch this. All right? So, a couple things before we begin. First off, the learning objectives for this video are the same as for the last one. In other words, you should know cellular respiration inside out, but this time with a whole lot more detail. If you feel up to it and you're ready for a challenge, let's go for it. Not going to change anything with our definition of cellular respiration. We're still going to use oxygen to help break down glucose and release energy in the form of ATP. We're still going to get CO2 and water as a waste. Net reactants and products of the process of glycolysis are still going to be essentially the same. But we are going to look at some details of the steps on how we actually get to pyruvate molecules. All right, so in glycolysis, we're going to break apart glucose, right? So we're still going to start out with glucose, C6H12O6. And what we didn't tell you about the glucose molecule is that a lot of times glucose is going to occur as a ring-shaped compound. But what we're going to do is crack it apart and stretch it out so it's more linearly shaped. And we're going to change it to a slightly different compound, still a 6-carbon sugar, called fructose. They have the exact same chemical formula, just the chemical bonds are arranged a little bit differently. So, the steps involved. We're going to go ahead and indeed invest 2 ATPs into this process. And, yeah, we're going to spit out 2 ADPs, but the free phosphates are going to remain with the new fructose molecule. So what we'll end up with is our fructose with 2 phosphates attached on either side. Now sometimes this is referred to as fructose bisphosphate or fructose biphosphate, and chemists like to kind of sort of number the carbons in the molecule so they know which end is top and bottom or which one is left and right. So it's going to be on the first carbon, or a phosphate's going to be attached to the first carbon, and then we're going to have another chain of linear carbons here, and a phosphate joined to the 6th carbon. So they'll call this compound 1,6, where the phosphates are, fructose biphosphate or bisphosphate. Clean this up a little bit. And with the help of some enzymes, what we can do here is take this fructose bisphosphate and whack it in half. Cut it in half. Essentially have 2 halves of a glucose molecule. And we call these phosphoglyceraldehydes, or PGALs. And we'll end up with 2 of those, each of them a half of a glucose. So if we clean this up a little bit, and we end up with something like this, right? Remember, each PGAL is essentially a half of a glucose. So instead of C6H12O6, each p-gal's got 3 of those original carbons, 6 of the original hydrogens, and 3 of the original oxygens. Well, using the appropriate enzyme, we're going to go ahead and strip off that phosphate and strip off one of the hydrogens. So, when we do that, we release some energy. Enough energy to go ahead and cause an ADP and a phosphate to go ahead and form ATP. And this little phosphate here is released. When it does, we can get an ADP to pick it up and become ATP. In addition, that hydrogen which is released, it can get picked up by NAD, just like we said before in our simplified version, and we have an NADH. Now the compound that's left over is still pyruvate. And we still have 3 of our original carbons, but only 5 of our original hydrogens, and 3 of those original oxygens. Now remember, whatever is happening to this p-gal here is happening to this p-gal over here. So again, if we clean this puppy up, we're going to end up with something like this. Right? Our ADPs and phosphates are fed in here. We can go ahead and have energy released, enough energy to go ahead and join these together, plus our free phosphate joins with this, and we've got ATP. Our NAD can pick up the hydrogen. We got our NADs, and we end up with pyruvates. But we've got 2 of them. So now we're ready for the next step, which is just pyruvate conversion, the first step of that Krebs cycle. And again, we're not really changing any of the bottom line events of pyruvate conversion. We still need all the same ingredients. We still end up with the same products. But again, just a little bit more detail. Remember, in the first step of pyruvate conversion, we wanted to go ahead and take that pyruvate molecule, which, well, each one has 3 carbons, 5 hydrogens, and 3 oxygens. And we wanted to strip off some hydrogen and some carbon along with some oxygen in the form of CO2. So here's the deal. What actually happens every time we want to go ahead and strip off these molecules in that form, what we're actually stripping off is something called a carboxyl group or a COO group. Note it has our missing hydrogen and our CO2. But our rule is we're always going to go ahead and strip this off as a COO. All right? So anytime we strip off carbon, we got to do it as a carboxyl group. That's why sometimes the Krebs citric acid cycle is sometimes referred to as the tricarboxylic acid cycle. So let's take a look at this in action. So that first step, when we want to go ahead and get from a pyruvate to an acetyl, we're going to strip off 1 carbon, 2 oxygens, and a hydrogen. But we do it as a carboxyl group. Well, from that carboxyl, NAD is going to come in, pick up the hydrogen, and become NADH. We're left with a carbon and 2 oxygens, which is just CO2. Now note that this happens twice because you've got 2 pyruvates. You end up with an acetyl molecule, just as we did before. And just as before, coenzyme A comes in, joins temporarily with it to become acetyl coenzyme A. Now we're ready for the next set of reactions, the rest of the citric acid cycle. Just as we had in the previous video, we're going to go ahead and completely destroy the rest of that glucose molecule, which is now just a couple of acetyl groups, and try and harvest some more energy and send the rest into the electron transport system. Now, we still have the same reactants. We got to start out with those acetyl coenzyme A's. We're going to need some hydrogen carriers, and once again, some chemist is going to be yelling at me and pulling their hair out because we're not really pumping in pure oxygen here. However, we're borrowing the oxygen from water. I keep this simplified version so that we can kind of sort of keep track of everything without going completely and totally bonkers. So forgive me for oversimplifying, but we're going to go ahead and look at just the oxygen's participation in this series of reactions. So before we gave you just the summary, and what we said was absolutely true, coenzyme A is removed, the acetyl fragment is going to be broken down, and we're going to end up with NADH, FADH2 carrying their hydrogens to electron transport, CO2 as a waste, and we do get a little bit of energy out of it. But now we're going to get into some detail. So if you really, really want to have some fun, well, here it comes. Well, we described that citric acid cycle as a cyclic set of reactions, a reactions that goes in a cycle. So we're going to end up where we start out. Okay? So a couple things to orient us. I've written the acetyl fragments in green here. That constitutes the original glucose. And remember, we got 2 of them, so I wrote them down twice. We want to go ahead and see what happens as each of the parts of those acetyls are broken down completely and totally. All right, so our acetyl fragments, we strip the coenzyme A off, and we join them to a compound which is actually a 4-carbon acid compound called oxaloacetic acid. And I'm not going to show the whole formulas. I'm just going to go ahead and leave this present as our starting molecule. Anyway, when each acetyl joins to oxaloacetic acid, we now have a 6-carbon compound called citric acid. Hey, this is why it's called the citric acid cycle. So what we're going to do is get this citric acid to actually lose a COO group. Remember our carboxyl, COOH? So we have to go ahead and somehow make, well, 2 COOHs because there's 2 citric acids. The only problem is we only have 1 oxygen to strip off from our acetyl group. Therefore, we're going to have to go ahead and borrow a couple oxygens. So oxygen has to be present in some form to go ahead and help us strip off that carboxyl group. Well, from that carboxyl group, we're going to go ahead and have the hydrogen stripped off by NAD. And of course, since there's 2 of those, we're going to need 2 NADs. And once that happens, we're going to have 2 NADHs. And we're left with CO2. So we're going to end up with 2 CO2s as as waste. And that's really messy, so let me go ahead and clear that up. Yeah, that's a whole lot better. Our oxygen is inputted, our carboxyls are released, NAD picks up the hydrogen to become NADH, and CO2 is our waste. Now we no longer have a 6-carbon citric acid, but a 5-carbon acid called alpha-ketogluteric acid. By the way, are all these names really, really super important? Well, I mean, kind of, but the main thing we want you to see is the steps that are involved in getting rid of each of the different parts. So we need to go ahead and go through this again, because we still want to get rid of this other carbon. And we got a carbon here and a carbon here. Now remember, if we want to get rid of carbons, we have to get rid of them as a carboxyl. But we don't have any free oxygen now to bind with the carbon. So we're going to have to go ahead and put some more oxygen into this. How much? Well, shoot, we're going to need 4 oxygen atoms to make CO2 here. So if we input the oxygen, we can now go ahead, strip off 2 of these carboxyls, and once again, the hydrogen can get picked up by NAD, and we're going to need 2 of them because we got 2 carboxyls, and we end up with 2 NADHs. CO2 is just a waste, so we end up with 2 more carbon dioxide, which are a waste. But here's the cool thing. When that last carbon is released, we can end up with enough energy to create 2 ATPs, or to convert 2 ADPs into ATPs. So, hey, we get a little bit more energy out of that. But unfortunately, it's not enough energy to keep a complex multicellular organism alive. So we got to harvest more energy, but first we got to get rid of all those hydrogens. Okay, this is where we left off, more or less, in just a cleaner version that you can actually see and actually read. We've pumped in our oxygen, giving us enough oxygen to get rid of a couple of carboxyl groups. We get rid of a couple more hydrogens, we create more CO2. And by the way, look at the CO2 that we've made here. We have 2 CO2s here, 2 CO2s here, plus the 2 CO2s that were given off during pyruvate conversion. That's 6 carbon dioxides. That's our 6 carbon dioxides given off as a waste in cellular respiration. Well, our 5-carbon alpha-ketogluteric acid has now been converted into a 4-carbon acid, but it's not the same 4-carbon acid that we started with. Succinic acid, that which is left here, looks a lot like oxaloacetic acid, except it's got a couple of extra hydrogens attached. And of course, these hydrogens are what's left of the acetyl fragment. So our next task is to go ahead and strip off the hydrogens. To do that, well, this happens enzymatically again. And each of these fragments, each of these succinic acids are going to give up a hydrogen, so we'll have 2 hydrogens. But for reasons which are better left unexplained at the moment, they're not picked up by NADs. NADs can't handle this at this stage, so what's going to happen is FADH is going to come to the rescue pick up the hydrogens, and that's how we get our FADH2s. Right? Now our succinic acid has, or each succinic acid has lost a hydrogen, and we end up with, again, a 4-carbon compound, but it's still not oxaloacetic acid. It still has one extra hydrogen. This 4-carbon compound, along with that last hydrogen from our acetyl fragment, is called fumaric acid. And all we got to do is get rid of that one remaining hydrogen to get back to our original oxaloacetic acid, and lo and behold, we actually complete the cycle and destroy the entire glucose molecule. So all we got to do is, again, use the correct enzymes, strip off a couple of hydrogens, one hydrogen from each fumaric acid, and at this point, NAD can come along, pick up the hydrogens, and become NADH. So our completed citric acid cycle looks something like this. And once again, I don't know if the names are super important to memorize. Probably the most important thing to look at is what happens to that acetyl fragment as you go from beginning of cycle to end. And also, the other important thing is what we get out of it. Some ATPs, waste, and all of those hydrogen carriers, which eventually you're going to need an electron transport. Our electron transport system works exactly as a summary, so I'm not going to really add a whole bunch in terms of detail. We're still looking at the mitochondria. We're still looking at the same reactants, the starting materials. The actual process becomes really complex. We'll talk about why in just a sec. The products are the same. And here's why it becomes like really complex. Remember that in order for something like NAD to pick up a hydrogen, you have to add 2 electrons. You might recall that from an earlier video, right? Well, where do all these electrons come from? And that's where things get really messy. We have to look at the interactions of that added oxygen and water. It becomes more messy than you'd ever want to handle in a short video like this. So we're not going to actually get into that detail here. If you really want to sit down and talk about it, corral one of your biology teachers, grab them by the collar, throw them in a chair or her in a chair, and have them explain your way through this, but you might go crazy before they're finished. Hopefully this flowchart looks somewhat familiar, and if you can explain your way through this flowchart, you're pretty good explaining your way through electron transport for the purposes of our course. So don't worry about adding more detail here, but you do want to go ahead and be able to explain how these ATPs are actually formed, how these ATPs are actually processed. In a previous video, we said that the NADH and FADH have their hydrogens kind of stripped by various protein complexes. Well, there's a cascading series of reactions in which this happens. Again, the details are probably not important, definitely not important in the context of this class. But needless to say, there's different protein complexes that handle different compounds. The net result is that both hydrogens and their electrons are stripped off and shuttled from place to place until eventually we can get a protein that will actually help combine the oxygen with the water. Again, don't worry about the details here. This is not something that you're going to have to know, even for extra credit. The actual process of ATP generation through a mitochondrial membrane, that is something that is kind of sort of important. So again, kind of a repeat. If you still remember this, you can skip the rest of the video. If not, it might be helpful to see it again. Once again, if this is the mitochondrial membrane, and we have a phospholipid bilayer, you have various protein complexes stuck in this phospholipid layer, and various channel proteins. Well, these various protein complexes are designed to go ahead and help remove the hydrogen from NADH and FADH2, converting them to just NAD and FADH, which can be used and reused over and over again. And the result is that you've got all these hydrogen ions accumulating on this side of the membrane. Since you have a high concentration of hydrogen ions on this side of the membrane, they will naturally move from areas of high concentration to areas of low concentration, providing they have the right channel protein. Channel protein in this case is in the form of ATP synthase, which allows hydrogen on through. And these things, using this method of facilitated diffusion, will go from areas of high concentration to areas of low concentration. Now, the movement of these charged particles gives us enough energy to convert ADP into ATP, and we've got some energy going on. Now, if we have 10 NADHs and 2 FADH2s, that should give us enough energy to go ahead and convert